Can you see the screen now? Here we are going to know karyotyping. We have discussed about this karyotyping on previous lecture. In very brief, we have discussed. Now we can know more about the karyotyping with the basics of the karyotyping. Okay? So let's know about some introduction. Human cells, as you know, the human cells have 23 pair of chromosomes, including 22 pairs of autosomes and one pair of sex chromosomes, giving total 46 chromosomes per cell, right? So 22 pairs of autosomes are there, you have to remember, and one pair of sex chromosomes are there, right? So in female, the sex chromosomes are 1x, 1x, so 2x are there, and in the male, there is 1x and 1y, so one pair. So it's always in pair, and together it gives a total of 46 chromosomes per cell. Now, karyotyping, what is this? So this is a technique that allows for the visualization and identification of human metaphase chromosome. So human chromosome during metaphase stage of the cell, what we do in the karyotyping, it allows us to visualize and identify the human chromosomes, right? And this technique can be used to assess the normalcy of an individual chromosomes. Means the individual chromosome is either normal or abnormal, so that can be determined by this technique. And also assay for various genetic disease. So this is one of the assay, one of the technique, which helps to determine various genetic disorders such as Down syndrome, Klinefelter syndrome. So these are the various syndrome, chromosomal disorder, abnormalities, and these abnormalities are usually determined by karyotyping technique. Here it is estimated that 1 in 156 live birth. So in every 156 live birth, one of the birth is usually defective. Okay, so they have some kind of chromosomal abnormalities, right? So in every 156 live birth of the baby, so one of the baby is usually suffer from the chromosomal abnormality. And to create a karyotype, chromosomes from a cell are stained and photographed. So what we can do, we can take a cell, nucleated cell, and we can take the chromosome picture, okay, and we can make a karyotype, right? So the chromosome picture is only possible if we have done the staining process, and then only we can do the photograph of the chromosomes, and after that we can create a karyotype, right? So to the photograph is enlarged and cut off into individual chromosomes while creating a karyotype, right? So when we have taken the photograph of the chromosome, so that photograph is enlarged. Then we do the cutting of the individual chromosome, and then we do arrangement of the homologous chromosome together. And here the homologous chromosome can be distinguished by length and by the position of centromere. Okay, so we can search the partner chromosome once we have done the cutting of individual chromosome. So we'll have a 46 chromosome in normal cases, and we have to search out the partner of each and every individual chromosome, right? And when we find the partner that we call as a homologous chromosome, when we search, then in the pattern of searching, or pattern of distinguishing the chromosome is based on the length of the chromosome, and the second basis is the position of centromere. So this length and position of centromere helps you to find out the homologous chromosome from each other. Okay. Next, a chromosome is divided by its centromere into short arm and the long arm. Short arm we call it as a P arm, and the long arm we call it as a Q arm, right? So here this is the centromere which divides the chromosome into to half. Okay, and the chromosome can be classified by the position of their centromere, that we have already studied these things. Okay, just we are recalling it: metacentric, submetacentric, acrocentric, and there is a telocentric as well. Okay, so metacentric, if its two arms are equal, then we call as metacentric, where P and Q become equal. Submetacentric, if arms length are unequal. Okay, so one of the P arm becomes shorter and long Q become a little bit longer, so they are unequal. And acrocentric, where the P arm is so short that is hard to observe but still present, right? And telocentric, I have not written here. Telocentric, there is no ER actually. So I have presented here three types of chromosomes based on the location of centromere. So centromere, you can see it is in the between P arm and the Q arm, so it's metacentric. Here the short arm P and the long arm Q is a submetacentric, and the acrocentric, where P arm is very small, which is not higher. This is acrocentric. So each chromosome has a characteristic size and shape in the normal cell. Okay. So if you see the normal cell, and if you see the each chromosome here in the normal cell, so each chromosome has a unique size and unique shape. Okay. Shape is depending on the centromere location, and size is what the length we are talking about. Okay. So the chromosome pair contains the largest chromosome. Okay, so chromosome in pair 1, okay, basically contains the largest chromosome, while the Y chromosome is the smallest. So if you talk about the grouping of the chromosomes, the first one is the largest, and the very smallest chromosome that is present in our cell is a Y chromosome. So Y chromosome is the smallest chromosome, whereas chromosome, which is the first pair, okay, the first number, it is the largest chromosome. And the chromosome of similar size and morphology are grouped together by letter. What we say, 7 groups are there. So the letter is used to group the chromosomes based on their size and morphology. So A, B, C, D, E, F, G. There are 7 groups of chromosomes which are divided based on their size and the morphology. So this is what we have seen here. The chromosome number 1, 2, 3 is grouped into group A. Okay, so group A, which contains chromosome number 1, 2, 3, and this is the largest chromosomes, and this is the metacentric chromosomes. Whereas group B is the set of chromosomes. Basically 4 5 chromosomes usually grouped into group B, and this is also larger chromosome, and this is basically submetacentric. And the group C here, there are 6 to 12 chromosomes are included in group C. So chromosome number 6, chromosome number 7, 8, 9, 10, 11, 12. All these chromosomes are grouped into group C, and the size of this is medium, and this is again submetacentric. Okay. Now group D, if you talk about, there are 3 chromosomes, 13, 14, 15. This is medium size chromosome, and this is basically acrocentric. Whereas group E chromosomes, there are also 3 chromosomes are there, 16, 17, 18. This is the short chromosome, and this can be metacentric or submetacentric, right? And the group F, which have 2 chromosomes, 19, 20. It has short chromosomes, and this is also metacentric chromosomes. So 19, 20 chromosomes are metacentric, and the short chromosomes are grouped into the group F. And the last, we have group G, where we have 21 and 22 chromosomes, and these 21 and 22 chromosomes are they are very short, and they are acrocentric. Okay, so this 21 and 22 chromosomes are, okay, they are very short and very acrocentric chromosomes here, right? You can see here this one, this 1, 2, 3, 4, 5, 6, 7 till 20. We have A group, B group, C group, D group, E group, F, and G group, right? And this XY is the sex chromosome is not placed here in any of the group which has kept separately here. But what you need to understand here, this is the normal karyotype, normal karyotype. Okay, where we can see all the chromosomes are in pair. Can you see all the chromosomes are in pair actually, right? And A has a three number of chromosomes, B has a two number of chromosomes, and the C has a six number of chromosomes. D group has a three number of chromosomes. E has a, again three number of chromosomes. F and G has a two number of chromosomes in A set. Okay, so this is what we can arrange the chromosomes, and this is the what the karyotype. Okay, this is the karyotype we have arranged by searching the homologous chromosomes. So this is the partner chromosome of this. Okay, so partner chromosomes are they are arranged together. Okay, partner chromosome of fourth is arranged together. Okay, so you can see this here. This is the image of this is the virtual image that we have prepared the chromosomes, where we can see the dark and the light band. Okay, light band and dark band. So based on the pattern of this chromosomes, we have also just represented this. Here the chromosomes are not in pair. Okay, there is no pairing of the chromosome. There is no homologous partner chromosomes here, but it has shown that these chromosomes are in a single. Okay, so here general principle. Let's know that how what is the principle behind karyotyping. Okay, so a blood sample is taken. What we can do? We can take a blood sample and uh, uh, white blood cells. Basically, the blood sample when we take, so we need to separate the white blood cells. Okay, so this white blood cell, basically lymphocytes, are grown in a special medium for 3 days. Up to 3 days, we need to grow these white blood cells, basically lymphocyte. Okay, and under the influence of mitotic stimulant. So the growth medium, the special medium, when we inoculate this white blood cells in this growth medium, this special medium, so there should be the mitotic stimulant chemical which stimulates the mitosis process. This is the phytohemagglutinin. Okay, phytohemagglutinin it actually is a mitotic stimulant which increase the mitosis process. Okay, so that the replication of the DNA can be enhanced. So this is the one of the chemicals which are kept into the growth medium. And after 3 days, what we say after not after 3 days, after 68 to 72 hours. Okay, when this time passes, then we need to add a mitotic inhibitor. Earlier we have added a mitotic stimulant. Now we need to add a mitotic inhibitor, which is called as colchicine. And this colchicine mitotic inhibitor is added so that it will stop the mitosis process and bring all the cells and the metaphase stage. Right. So all the cells will come to the metaphase stage. And after treatment by hypotonic solution, so when we go for after addition of this colchicine, we need to treat the cells using the hypotonic solution, which causes swelling of the cells and allow the dispersion of chromosome within the cell membrane. Okay, so within the cell membrane, within the cell, the chromosomes are dispersed because of the swelling of the cell when we keep all the cells in the hypotonic solution. Right. Now, what happens? We need to do the fixation after that. Okay, fixation is used as a wash solution. Okay, which lyses the remaining red cells. If there is a red cell in the sample, in the nucleated cells, which can be lysed by the fixatives, and it will also remove some chromosome proteins. Some proteins which are used in the making of the chromosome that can all be removed during the washing process. And after we do staining, where staining is done can be done by Giemsa stain. Okay, so there is a mistake in the spelling. It's STAIN. Okay, Giemsa stain can be done so that the chromosomes can be colored. So chromosomes can be microscopically observed and evaluated for the abnormalities. So we can evaluate if there is abnormalities in the chromosome number or chromosome shape. Okay. What materials do we require actually? The materials that is required for this karyotypings are we need heparin blood. We need a special medium, what is called karyotyping medium, which should contain 5 hemoglobin. Okay, 5 hemoglobin. This is the name of the medium, RPMI 1640. This is the name of the karyotyping medium, which also contain 5 hemagglutinin, which is a mitotic stimulant. Now we need an incubator with the 5% of carbon dioxide supply, and the temperature should be maintained at 37 degrees Celsius. And we need a coalcimate solution with the 10 microgram per ml, and we require potassium chloride hypotonic solution, which concentration is 0.05 molar. We require a fixative solution. This is the how we can prepare. We can prepare 3:1 ratio of methanol is to glacial acetic acid, right? And we need a gymsastin solution. Size microscope. So these are the requirements should be arranged before proceeding to the procedure of the karyotyping. What is the procedure? There are different steps are there, so you need to remember what are the steps. Okay, here the first you need to inoculate the 0.5 ml of whole blood into a tube. So you'll take a tube, you will add 0.5 ml of whole blood, and then we'll add a 10 ml of karyotyping medium. Okay? So just we are putting the whole blood, 0.5 ml of whole blood into a 10 ml of the karyotyping medium. Then after that, we need to incubate the tube in incubator. This is the requirement: 5% carbon dioxide and temperature 37 degrees Celsius. So this is the requirement for incubator. The incubator should be supplied with the carbon dioxide and should be maintained at 37 degrees Celsius. So we can do the incubation for approximately for 72 hours. Then after that, after a total of 69 hours, when there is a 69 hours of total incubation, so we can what we do that from seeding we can add 100 microliter of coalcimate. Okay, so at the time of 69 hours we can add coalcimate this much amount, 100 microliter. Okay, in each culture tube you can add. Again we can incubate the tube at 37 degrees Celsius for additional 20-30 minutes. Right, then we need to spin, means we need to centrifuge for 1500 rpm for at 1500 rpm for 7 minutes. So this is the centrifugation. Then after centrifugation, what do we get? We are supposed to get a supernatant. So we should remove the supernatant and we will resuspend the cells in 5 ml of hypertonic solution. So this is the potassium chloride pre-warmed at 37. This should be warmed at 37 degrees Celsius firstly. Then after that we can take 5 ml of this solution and we can resuspend the pellet what we have got after centrifugation, right? And then we can incubate again for 50 minutes, and then we can add drop by drop 1 ml of fresh ice cold fixative. So whatever the fixative we have prepared, the 3:1 ratio of fixative methanol and glacial acetic acid. So on drop by drop we can add into the solution, and we need to do vortexing also, mixing also. So again we need to do this centrifugation for 7 minutes, and we need to remove the supernatant, and again we need to add drop by drop of this fixative approximately 5 ml, right? And leave at 4 degrees Celsius for 20 minutes. So what we are doing here, we are adding fixative because we're trying to remove the cells other than what the cells we are required. We should identify lymphocyte. We should isolate lymphocyte. We should separate lymphocyte, and we should remove the other cells. So during this 2 times 3 times process of washing, all the cells and other proteins are actually washed out. Okay, so we need to repeat 9 and 10 steps. This 9 steps like centrifugation, then we can remove the supernatant, you can add this fixative, and again we can repeat. This process can be repeated at least for 2-3 times. Okay, so after repetition, we should centrifuge, we should discard the supernatant, and we can resuspend the pellet in 1.5 ml of fresh fixative. So what the fresh fixative, how much we have taken? 1.5 ml, and after that, drop by drop, approximately 4 to 5 drops of this from high of approximately 30 centimeter onto a clean slide. So from what it is written here, at 4 to 5 drops we can take it on a clean slide, and we can blow carefully on the drops of the spreading, for the spreading them on the slide. Means we have to just make a smear. Okay, we have to add 4 to 5 drops of this cells, and then we can blow carefully on the drops so that it can spread on the slide. Okay, uh, and then put the slides on 45-degree heated plate for 2 to 4 minutes. This is the what required, that at 45 degrees Celsius heated plate, we need to put the slide approximately for 2 to 4 minutes, and then we need to again heat the slide to 60 degrees Celsius for overnight, or 90 degrees Celsius for 90 minutes. Then we can place the slides and float them with the Giemsa stain solution for 8 minutes. So this is the staining procedure we are doing, that we need to stain Giemsa stain for 8 minutes by covering the smear, and then gently rinse the slide in distilled water and air dry. And now we can do it, the microscopy procedure. Okay, we can observe at 10x, 40x, or 100x. And then after that, finally, we need to focus on a special one cell, and then we can take a photograph. Then we can print out the photograph. We can cut it. You can cut each chromosomes from the photograph, and we can arrange the chromosome according to the size in the position of the centromere. So this is what we need to do is. So here, this is what happens. This is the summary of the steps. Okay, that we have taken the blood sample. Then we have removed the RBC. We have added the RPI medium. Okay, we have grown till 769 hours. Okay, in the RPI medium that contain RPMI medium contain PHA. Okay, phytoagglutinin. Then after that, we can add colchicine. Okay, we can add hypotonic solution like water, and we can further we can do the washing process, and we can do the staining, and then we can put on the slide. We can stain it. Okay, and finally we can take a photograph. Okay, and this is the photograph which is seen here. Okay, so this photograph, now we can cut it out the individual chromosomes, and we can arrange in a in a homologous chromosomal pattern that we need to arrange according to the size, according to the centromere, right? And here what we see, that one of the cell is showing chromosome inside the its cytoplasm. So we need to take the photograph, and we need to cut each and every chromosomes. Okay, and we need to make this kind of pattern. Okay, 1, 2, 3. So this A, B, C, D, E, F, G. So this is what we have done the karyotyping. And here, this is what we have written. There are 46 chromosomes are there, and the pattern of the chromosome we have written with the XY, means this is the male chromosome, male karyotype. Okay, male karyotype of male patient. So this is normal male karyotype, and this is a normal female karyotype. Okay, where no, this is also the male. This is also the male. Okay, because there should be the X. There is a mistake. Okay. So now you can see this is the abnormal karyotype, where we can see all the chromosomes are in pairs except chromosome number 21, where you can see there are three 21 chromosomes. Okay. So this is the Down syndrome, where there is a trisomy of 21, right? Now you can see this karyotype, where we can see chromosome number 13. There are 3 in number. There is 3 number. Okay, so 47 XY, and we can write plus 13. Okay, this is the pattern of writing the interpretation, the result, and the previous also you can write in the same way. Okay, so 47, because the total chromosome is not 46, it's a 47, and this is XY, and here with comma we can write that which of the chromosome is extra. So this is plus 13 is the one chromosome number, one chromosome is extra in 13 chromosome number. So this is the karyotype of syndrome. What Patau syndrome, where there is a trisomy of 13, and this is what we can see. The here it is highlighted. The 18 chromosome is 3 in number. Okay, and this is what we have written the karyotype as. Total is 47, which is XY, and with comma we have written plus 18, which means there is one additional chromosome of extra chromosome of chromosome number 18, and this is a Edward syndrome. Okay, and here we have 2X and 1Y chromosome. Okay, so here total chromosome in the cell is 47, because one additional chromosome X is there. So we write XXY. Okay, and here this is a Klinefelter syndrome, and you can see here that one of the chromosome is missing. Okay, it's not Y, not X. Okay, so we have total number of chromosome is 45. Okay, and we have written X. So 45X. So the sex is not determined because it's not either double X is not there, and only single X is there, and Y chromosome is also missing. So we write 45X, and what is the type of syndrome? We call it is a Turner syndrome, where there is a deletion of one sex chromosome. It has only one X chromosome. Okay, and this is what we have studied, the karyotyping and the chromosomal disorder. We have seen some karyotype also. Now coming to the discussion, if you have any question, just please ask me. Okay, is there any question?